And let's welcome Nicolas Stransky, the Corporate VP of Data Science of Caris Life Sciences. Nico, the floor is yours. I know that there is a slight change in the titles of the slides, so I'll leave it to you to introduce the title.
Thank you. All right, so I'm going to take us in a slightly different direction today than the first couple talks. The reason for that is because drug discovery spans really a lot of topics, especially in industry, where in drug discovery, first we need to understand what the biology of the disease is, what potential targets could be, all the way down to identifying a patient population. So who are we going to treat? How are we going to find those patients? How can we predict whether they're going to respond or not to a particular drug? And of course, drug discovery also includes coming up with the drug or identifying the relevant targets and the structure of those targets. And so what I want to focus on today is aspects that relate to our use of genomics and diagnostics and molecular data to better understand patient populations and predict drug response and outcomes. So my name is Nicolas Stransky. I lead the data science at Caris Life Sciences, which is a cancer diagnostic company based in Texas and Phoenix, Arizona, and also here in Cambridge, where there's a data science team that I lead. So to start with a little bit of additional context, 30 years ago, the first targeted therapy was invented, imatinib against ABL, which is a kinase that drives acute myeloid leukemia. And following that and that revolution in the care of that disease, there was an enormous effort to identify more of these targets, more kinases and other kinds of targets that could be treated directly with the drug in a targeted fashion, meaning precisely inhibiting what's causing the disease. And shortly following that revolution, there was the rise of what we called next generation sequencing, driven by multiple companies who really revolutionized the space. Not with that exact sequencer in the 2000s, but still there was an enormous revolution in the generation of genomic data, which led to multiple improvements in the field. One, much better molecular characterization of diseases, cancer and other diseases. That led to better target identification, but also gave us the tools and the data to identify those patient populations. And then because of that enormous rise in the amount of data that was generated, genomic data, we needed better tools to analyze those data and tease out what could be driving disease, what could be specific targets, and what could be subsets of the patients that might respond to a drug, and AI or ML are extremely useful for that, and that's what I want to talk about today. So following that rise of the use of sequencing, we went from a symptomatic diagnosis. There's a mass, there's a different appearance of a certain condition, to really profiling molecularly cancer and other diseases to understand what genes are involved, what mutations, any kind of other alterations are involved, so that we can very effectively match a drug in a patient when it comes to either the response or the outcomes. The drug space or the treatment space in cancer comprises a lot of drugs that are associated with a particular diagnostic, so the so-called companion diagnostic, which require that we identify the precise molecular alterations that would allow the doctor to prescribe a drug to treat the disease. And in this slide are depicted multiple of these targeted inhibitors. So they're mostly small molecule inhibitors on those slides, together with the kind of molecular alterations that need to be detected in order to prescribe the drug. So that's really a combination of the availability of the drug and the ability of the physicians to identify those patients. And that's why the field of diagnostics is so important because these are not drugs that can be prescribed to all patients for a particular cancer. These are drugs that need to be prescribed to a patient when they have the particular alteration. And that's the enormous challenge of identifying those patients, because it requires very advanced profiling techniques, very costly profiling techniques for those cancers that span multiple technologies. And the reason why I have this diagram in the middle is because we're spanning DNA sequencing, RNA sequencing, protein profiling through immunohistochemistry, and other technologies, and all of these need to be done most of the time on the same sample to identify which drugs might be prescribed to a patient. So with that context, Caris is a diagnostic company. We're mostly a sequencing company generating an enormous amount of sequencing every day, 24/7. Probably the biggest sequencing company. There's 50, actually many more, 50 NovaSeqs down in Arizona to perform all the sequencing, 50,000 cancer samples per month. That's tissue samples. There's also liquid profiling from blood biopsies and hematological malignancies profiling soon. As I said, there's labs in Arizona, in Texas, and data science in Cambridge. So the kinds of diagnostic that need to be performed employ these various data types that I mentioned before: DNA sequencing, RNA sequencing, which allows us to identify certain fusions or certain gene expression. DNA, of course, allows us to identify point mutations, also fusions, and copy number aberrations, and other kinds of aberrations that are important for the prescription of drugs. And then, of course, through protein, we can identify certain markers that would be overexpressed or present and allow us to prescribe certain drugs. So all of that is very important for diagnostics, but it's also very important for drug discovery. And it enables R&D, identifying new targets, identifying new patient populations. And the enormous amount of data that's available to us at Caris and elsewhere enables such activities at the beginning of drug discovery. So that's what I want to focus on today. So let's start with an example of one such diagnostic that has existed for 20 years, Oncotype DX. It's a diagnostic that's used in certain breast cancers and predicts the likelihood of recurrence after treatment with a certain therapy. It's based on the gene expression of multiple genes that are profiled through RT-PCR, so it's an old technology. It's robust, but it's been around for decades. And then there's a very simple score that's calculated that gives a numerical number that allows to determine the next course of treatment. And there are multiple such signatures of scores that exist in the industry, and this one seems pretty simplistic, although its discovery was not. But there's probably better tools that we can employ today through the enormous availability of data that's generated every day by many companies, and through the use of machine learning and other techniques to come up with better signatures. So with that, we need better signatures. We need better signatures because because we have the means of doing so through better data and better techniques, but also because the current ones don't work that well. And there's not perfect matching between drugs and patients. And I want to start with a quote that seems a little grim, but is true. Most drugs fail most patients most of the time. Most drugs are not going to work on a particular disease. Most patients are not going to respond to a particular drug, and when they do, they're actually sadly going to oftentimes recur and progress in their disease. And that's even true for the recent generations of therapies, so-called targeted therapies that are much more tailored to an individual's tumor. So we need better signatures because we need to match much better the drugs and the patients. On top of that, drug resistance, which is a phenomenon that either occurs right when the patient is given the drug or that a cancer evolves to have and not respond to the drug anymore. Drug resistance is not due to any one mechanism. It's due to a multitude of mechanisms, and sadly that means that just profiling one gene or one part of the genome is not going to tell us whether there is resistance or not. We need to look everywhere to identify or understand drug resistance, and that's also a necessity of employing larger and larger data sets. Methods that are going to maximize our chances of understanding drug response, drug or targets are most likely to be helpful. That use the methods that use both genomics, transcriptomics, pathology images, other kinds of clinical information. So using as much information as possible is going to teach us more about what's happening in the disease and which patients we want to identify. And finally, of course, machine learning is highly suited for all these kinds of questions. So with that, jumping into the first principle of what I want to talk about, how do we use machine learning to establish a robust data set to enable R&D? And by that I mean identifying better targets, identifying cancer drivers, and identifying patients. So 10 years ago, this paper was published here at the Broad, actually, and made certain predictions about what kind of data are necessary to better understand multiple kinds of cancers, down to the percentage of, down to 3 to 1% of prevalence of certain mutation. And so what it did is, depending on the mutation frequency across a variety of cancers that are depicted at the bottom, it made predictions of how many of these cancers need to be sequenced in order to identify a mutation with a prevalence of 10, 5, 3, 2, and 1%. And as you can read on this y-axis, these are staggering numbers. They were completely improbable numbers 10 years ago, because by that time, we were, or a lot of people were in the middle of the analysis of already a very large project for cancer sequencing, the Cancer Genome Atlas, or TCGA. And the number of cancers that was sequenced in the TCGA was altogether between 10 and 15,000 across all of these cancers. And so thinking that we would need one day several thousands of, we would need one day to sequence several thousands of samples for one particular cancer was not imaginable at the time. Of course, we knew that with the progress of sequencing, that would come one day. And that's now. We're now at the cusp of achieving that scale to understand and discover new cancer variants because we're starting to have the numbers. Of course, there's one important aspect here, which is that we need to focus on somatic mutations. Somatic mutations are those mutations that occur specifically in the tumor and that are not polymorphisms, meaning that they're not part of our normal genome. So somatic mutations, they can be discovered when we're comparing a tumor genome with a normal genome. But in the course of the data that we generate at Caris, we're only sequencing the tumor, and that's for multiple reasons. One is for cost reasons. It would cost twice more to sequence both the normal genome and the tumor genome, but also because logistically it's much more complicated to deal with two kinds of samples, one liquid sample from blood and one sample from one solid sample from the solid tumor. Of course, it's possible to sequence both tumor and normal, but it's more costly and impractical. So then we're left with a question, which is how do we constitute a data set for discovery that is purely composed of somatic mutations? So the first quick example I want to talk about is a technique that we devised that we called SOM-alone to identify the somatic mutations in tumor. So it's tumor-only somatic germline classification. The goal was to apply it to the collection of 500,000 cancer cases that we have, that I'm going to talk more in a couple of slides. But we used to devise and train this method, we used a smaller data set of matched cancer tissue and normal blood sequencing of a size of about 300 samples. And that gave us the ground truth of what somatic mutations versus germline look like. And long story short, this algorithm is using two main sources of information. One, the frequency of polymorphisms in the normal human population, and the allele fraction of those mutations in the tumor that was sequenced. So in all our cells, there's multiple polymorphisms, about 1 per 1,000 bases. And in the case of heterozygous germline mutations, they're present in about 50% allele fraction. So the polymorphism is about half the reads in the sequencing data, and that's in all our cells. And in tumors, or in tumor samples, there's a mixture of both normal cells and tumor cells. Normal cells are the blood vessels and fibroblasts and immune cells that are infiltrate the tumor. So there's always a mixture of normal cells and cancer cells in the cancer sample that we're sequencing, which actually works to our advantage. Because when then we look at the mutations that are present in cancer cells, they're going to come at an allelic fraction or a B allele frequency that is lower than that of the normal cells. So we're going to use that kind of information to potentially detect which mutations come from the tumor and which come from the normal cells. And so to illustrate this, what I'm depicting here is chromosome 1. So these are the genomic coordinates of one chromosome on the x-axis, and on the y-axis are all the mutations that we can find in the tumor sample. So all the dots are the mutation with their B allele frequency, meaning like how frequent is that mutation in the sequencing data. And that's for one of the samples that was in the ground truth data set. So there's matched normal, which allows us to identify which of these mutations are somatic in red, because they're not present in blood, and which ones are germline in gray, because they are present in blood. And the principle that I just talked about one slide ago, that tumor mutations are going to be preferentially at a lower B allele frequency than the germline mutations, is generally true. The red dots are most of the time lower than the gray dots. And that's information. It's imperfect and incomplete, but it already gives us information. So when we make a model to use that kind of information to predict whether the mutations are germline or somatic, there is a relatively poor performance in that exercise, which is expected, but still important. So second, there are actually really good databases of germline events that exist, and one is Nomad. That's also a project from the Broad and multiple other centers that catalogs all that attempts to catalog all the polymorphisms in the human population. For that, they're using a data set of 800,000 exomes and genomes, and look at the frequency of every single mutation in that data set, and that allows to make this database of polymorphisms together with their frequencies, and that's extremely useful for research. So using that, we can compare all the events that we find in tumors with that database of germline mutations, and estimate based on the frequency in the population whether that's going to be likely a germline event, meaning present in normal cells, or a somatic event. And if mutations are frequent in the population, and we find it in tumor, it's likely germline. If they are not present in tumors, in the general population or at a very low frequency, it's likely somatic. So that's a second source of information that we can use, and that gives us better performance in predicting from tumor alone whether we have a germline or somatic event, but still not perfect. And combining both, we can achieve better performance in predicting whether we have somatic or germline events. And it can actually get really either good precision or a good recall. And what this allows us to do is then go back to the entirety of 500,000 exomes from tumor only and sort all the mutations and focus precisely on somatic events. So why is that important? It's important because then we can go back to the question that was identified 10 years ago of how many samples are needed to identify driver mutations in a series of cancers depending on their mutation frequency. And now we have the numbers and we have the somatic mutations in those tumors. And these are numbers from the collection of data that we have assembled or are assembling at Caris through diagnostic testing. And there's 12,000 melanomas, more than 70,000 lung cancers, and we're like well into the numbers of tumors that are needed to better understand those tumor types and now focus on identifying new cancer drivers. And that's work that is happening right now. So I don't have the second part of this story, but at least we are able to analyze those data of an extremely large scale to constitute better data sets for R&D and drug discovery. So next, I want to talk about a second aspect of the use of machine learning, or actually there's not a ton of machine learning in that short section, but it's really perfecting the data set that is going to be used for R&D. In this example, I want to talk about an effort from last month, actually, where we reprocessed close to 400,000 RNA-seq samples. So like I described at the beginning, for every single tissue sample that comes through the door at Caris, we're generating a whole exome sequencing and a whole transcriptome sequencing, and also a lot of IHC and so a lot of digital pathology images. And we needed to go back to the entire data set for RNA-seq to improve certain aspects for the purpose of R&D, and went back to raw data and reprocessed 400,000 samples in a matter of a couple of days. And that was really a notable effort. That was both a scientific and also engineering feat to generate that amount of data in such a short time. And for those who are interested in the stack employed here, I'm writing it here, but basically in AWS, we build up an infrastructure to deal with that amount of data. And it's important to do so and go back to raw data, because in a diagnostic test, there's evolutions every day of how we can improve the workflows, how we can generate better data. And so year after year, the data change a little bit, morph a little bit, and when we want to go back to all the data, we need to take steps to harmonize and make sure that we don't have these effects and drifts in the data so that they can be analyzable. And the advantage of that is, maybe to go back to one of Pat's points in the intro, what is needed for ML is data, and the data is there, and it needs to be put in a shape and a form that is usable then for drug discovery and the kinds of analyses and machine learning we want to do with those. So briefly, the RNA-seq data generate expression data, so the expression of all the genes in the genome. Those expression data can be used to compare various tumor types, and there's a lot that can be done with those. That's also something that is starting. Back in the days of TCGA, we were using 10,000 samples across 10 to 15,000 samples across all tumor types. Here, there's close to 400,000. There's even more in the collection. This can be represented. So all these transcriptomes meaning gene expression, can be represented in this t-SNE plot, which is typical for folks who work with single-cell data. With single-cell data, it's not unusual to see 250,000, 500,000, or 1 million cells across a few samples. Here we're not talking about cells. Each dot is one sample, and there's close to 400,000 samples depicted here across a variety of solid tumor types. As we would expect, the samples cluster by cancer type due to the expression differences between these samples. I forgot to do so earlier, but this work and then the previous work that I showed on some alone is the work of multiple members of the team, Noah Spies, Laura Richards, Mukund Varma, Christian Fresh, Greg Demarais, Brady Gilg, and others in the data science team. I'm only going to barely scratch the surface of what can be done with these kinds of expression data here, but I want to give an idea of why that's important for drug discovery and what kind of features we can distinguish in those kinds of expression data. So here's colorectal cancer. There's close to 50,000 colorectal cancers in this data set that are depicted here in brown in this t-SNE plot. And we can start distinguishing some features that tell us about the biology of those cancer samples. So everything in brown here is colorectal cancer, including this little island here and this little island there. And when we color the data set now by where the specimen was sampled, so where it was coming from in the body, so either the liver in the case of a liver metastasis or the lung in the case of a lung metastasis, we can see that this has the site of the biopsy has an influence on the transcriptome of those cancers, and so that's why they belong to this slightly different islands in this t-SNE plot. So anyway, that's really just the surface of those data. As you saw, this massive data set was just generated last month, and so this is very new, but now constitutes the kind of data set that can be used for R&D, both from mutations, but also transcriptome data, and we need to dive in, not just us, but also partners of Caris that are working on this data. So next, I want to talk about the application of machine learning to improve treatment decisions. That's one where we, as I said, we need to do better with signatures. We need to do better identifying patients and identifying the disease characteristics that are going to guide treatment decisions. And for that, I'm going to focus on this aspect of the diagnostic test that is available to patients who are profiled at Caris, where we can use the variety of data that are generated through the diagnostic test, so the whole exome sequencing, the transcriptome sequencing, and the IHC, through a model that identifies the exact tumor type of the tumor that was profiled. And that's work from Anthony Hemstetter and Hassan Ghani in the team. So the purpose of this mainly is to identify the primary tumor for the case of cancers of unknown primary, which are a kind of cancer where the doctors cannot identify which cancer type they are dealing with. And that's very important because the outcome for cancers of unknown primary is very poor, about a year, less than a year, because the doctors cannot determine which drugs would be best suited for treating that cancer. So they revert to sequencing to learn more about the cancer, which can tell what kind of mutations exist in those tumors, but mutations alone do not drive treatment decisions yet. It's really important to understand what kind of disease we're dealing with so that they can follow standard of care. And for that reason, identifying or being able to identify the primary origin of these cancers is very important, and that's the goal of this GPS test that's performed on the data generated. Very briefly, it's multiple models that are using the whole exome data that go down the tree here and classify every time which branch of the tree to go through and go through additional levels of granularity through the oncotree. At the end, there's a prediction for which cancer type out of 90 cancer types can be is predicted to be the cancer of origin. It's been trained on about half of the available data at Caris and then tested on more cancers of unknown primary and with known primary origin and validated on prospective cases. And that's the aspect of validation, just as a brief aside. Validation of diagnostic testing is an extremely complicated task where the bar is extremely high to submit those kinds of tests and performances to the FDA so that there's really the certainty that the results can be actionable by the doctors and by physicians. So every time we're talking about these kinds of predictions and machine learning or not from diagnostic data, we also have to understand that there's a very high burden of proof of can that be applied to a patient. Would I use that test and trust the result for my patient? To close the loop a little bit, and this test has been applied to the TCGA data that I talked about earlier. So like I said, there was this here only 8,000 of the 10 to 15,000 tumors in TCGA, but we could validate it with a very high performance on those TCGA data, which of course have known tumors of origin, but with masking the tumor of origin and applying GPS onto this data, we could repredict what the tumor types were, and that was a very good validation of the performance of that test, and robustness to the source of the data, which is really important. It's very robust to the sites of metastasis. As we discussed, the transcriptome data is going to be slightly different, whether we have a primary tumor or a metastasis, depending on which organ. So those are aspects that are going to be really important when applying this test, so that we're not making false calls. But it's robust to that. So with that, I want to give a brief patient highlight, because this was really notable, and we actually have many examples of how that kind of test applied to cancers of unknown primary is really helping treating patients. So in this instance, I'm talking about a male specimen submitted as a renal cell carcinoma, so like a cancer in the kidney, arising in a transplanted kidney. And so first thought is probably kidney cancer, but GPS favored the gynecological origin with 99% probability and histology of serous carcinoma, which of course is a female cancer, but so that raised questions from the beginning. When comparing the SNP profile of the tumor and that of the benign kidney around the tumor, it showed that there was a negligible chromosome Y copy number, which raised additional questions, but also that the tumor matched the transplanted kidney. And in fact, of course you guessed, but this was not renal cell carcinoma. This was a ovarian cancer that was already present in the transplanted kidney at the time of the transplant, and the oncologist was updated, and the oncologist was made aware, and they updated the therapy accordingly, and that led to much better treatment for that person, which otherwise would have been treated for kidney cancer and probably would have not received the right treatment. That's the kind of treatment decisions that can be informed by the use of ML on this diagnostic data. So next, I want to talk about the use of ML for predicting biomarkers and patient outcomes. And that's where we're going to go in a slightly different direction here with using the digital pathology data at Caris. So there's two kinds of pathology data that we're generating for every single case that comes through the door. One is H&E, so hematoxylin and eosin, And the other one is IHC, so immunohistochemistry, where we're staining the slides for a particular protein that binds an antibody. This is used to confirm diagnosis. So the pathologists are looking at these slides and identifying the shape of the cells and the histology to confirm that we're in fact dealing with the cancer type that we thought. It's also used to determine the percent tumor nuclei, so the purity of the sample and where the tumor areas in the slide are, because we're microdissecting every single sample that needs to before sequencing. What that means is when sequencing samples, they're first, the block, the FFPE block, so the paraffin-embedded block of the tissue is cut in thin slides. Those are put on a microscopic slide. A pathologist is looking at the tumor, identifying where most of the tumor cells are, and then microdissecting that area to go to the sequencing assay. And what this the consequence of that is that it creates much better data for diagnostic, because we're not sequencing a bunch of normal cells. We're focusing the sequencing on the tumor cells when needed. If the tumor is already pure, it just goes straight to sequencing. So that's great for diagnostic. But it's also great because these images are all digitized, and there's a ton of information in them that we can use machine learning to leverage. Very briefly, as I said, all the images are digitized, and so that's use of robots that are picking the slides, putting them in the scanner. That's all uploaded to cloud and various tools that allow the pathologists to look at them. There's automated scanning and everything. So there's really a massive scale here. As you remember, there's more than between 15 and 20,000 tumors per month that are going through the labs. All this data go to our cloud storage and can then be used for various efforts. Of course, at the beginning, we need to go from a scanned image that contains some labels, some pen marks, some that has various sizes and shapes and everything, and detect all the areas that we would want to focus on in a computational fashion. So where's the foreground, which areas are in focus or out of focus, are there artifacts, are there labels we need to remove, and finally dealing with the images that's going to be interesting for our data. There's close to 4 million images, both from H&E and IHC, that represent 350,000 cases, and we're digitizing everything that comes through the door today, but there's millions and millions of images that are that have not been images in years, that have not been digitized in years prior, that are also being digitized. So this is a growing data set of a massive scale already. So like I said, there's a ton of information in these images. Here, two examples of breast cancer, one with the CDH1 mutation, and the other one with a fusion gene, so a translocation that leads to the overexpression of this gene NTRK. And they look massively different, and that's obvious to the eye, that's obvious to a pathologist. But in some instances, the differences are not obvious to anyone, and neither the untrained or the trained eye, and that's where we want to use computer vision to help us make those distinctions. We can employ a bunch of models. I'm going to give two examples, all for the purpose of modeling tumor types or drug response or various aspects of tumor biology. So first use case, let's say we have a clinical trial against for a kinase inhibitor, ROS1, against ROS1, and we want to identify 100 patients that have a fusion in this ROS1 gene. The problem here is that the prevalence of that fusion is about 1% of lung cancer. And if we want to recruit those patients and identify those that harbor that fusion, we need to sequence the genomes of 100 patients to find one. And so if we want to identify 100 patients, we need to sequence 10,000, and that has a staggering cost. If it's several thousand dollars to sequence one sample, you can imagine the cost of sequencing 100. So how can we use ml to train a model that enriches the cohort so that we don't have to sequence so many. So that's what we did. We briefly used a CNN to train a model on a lot of our data to identify these ROS1 cases. And here you can see that the enrichment that we're able to achieve. So then running it on unseen cases, if we, instead of naively sequencing 10,000, if we run the model on 25,000 H&E slides, we can identify a short list of 1,000 cases to sequence, so already a decrease in 10,000, a tenfold decrease in the cost of the sequencing. And further, if we run it on 30,000 H&E cases, we only need to sequence 300 cases to find 100 that would be enrolled in the trial. And so it's that kind of machine learning that can help drug discovery, because it's going to help the clinical trials for a particular drug and definitely benefit the patients. That's work by Tommy Boucher, Mukund Varma, and Ming Cheng, and others in the team. Briefly, because I think I'm close on time. Okay, a couple more minutes. Some learnings, we need to account for the various sizes of the data and its heterogeneity. There's class imbalance, so we need to account for that when we're training the data. Like ROS1 fusions were 1% prevalence, so it's really hard to train data at 1% prevalence. We need to take that into account and then make sure we're not overfitting, and then we're dealing with data that have multiple resolutions. So like a microscope, we can either look like a large field of view or very narrow, and we can actually employ multiple magnifications at the same time to train better models to see the overall structure or really the cellular structure. The next example, which I will, it's several slides, but I'm going to abstract from it. So we wanted to provide an alternative. We're working on providing an alternative to the test I talked at the beginning, Oncotype DX, which is an RT-PCR test based on roughly 20 genes, gene expression. That's costly to generate, and given all the data that exists today, we can probably do better. And to decrease the cost, one aspect of doing better is to start from H&E slides, so digital pathology slides, instead of expression data. For that, we're using data from a clinical trial where, simply put, there was a breast cancer diagnosis, then followed by 5 years of treatment with endocrine therapy. And at the end of the 5 years, some patients were put on a placebo, and some other patients were put on additional endocrine therapy for 5 more years. And then there's a readout for 10 years. So really, we're looking at 15 years from diagnostic to the end of the follow-up. And we're going to use data from day 0, so at the time of diagnosis, to predict whether there's going to be a recurrence 10 to 15 years down the line, and whether those patients would benefit or not from additional treatment with the endocrine therapy. Computer vision model, and we're extracting features from the slide, cut into tiles, so a number of features using a transformer, predicting a score, whether there's high risk of recurrence or low risk of recurrence. And it works. So between the predicted low risk and high risk, we can see that there's a large difference in this survival curve when looking at all patients. So I'm not even focusing on whether they used additional endocrine therapy or no. That's done from the images only, so no other information, or incorporating some of the clinical data into the model, such as type of the tumor, whether they received surgery, and other things like this. Now where this becomes interesting is where we look at, where we bin the data, whether the patients received additional endocrine therapy or received placebo. In the low risk group, very, very low risk of recurrence. In the high risk group, we see a difference between those two groups. But what this tells us is that these patients here probably don't need to be treated. It doesn't provide an advantage, and that's a very, very important information to not overtreat patients. This can be compared to the results of these other tests. So another test that does the same is the breast cancer index. We're achieving very good, if not better, performance, and that's really the basis of proposing better diagnostic, cheaper diagnostic with modern techniques and that are going to help patients. So with that, I would like to thank you all for your attention and take any questions.
We are a little over time, but I would be happy to take at least one question for Nicolas. If no question from the audience, I have one question for you, Nico. As you have been working in this field of data science and in cancer, you talked about RNA-seq data, pathology data. What would be one data type that you wish we had more and need to produce more, which will help developing machine learning?
Proteomics, large-scale proteomics. Okay. That's awesome. Very, very important. Today, it's really hard to generate large-scale data for the expression of a variety of proteins. There are techniques that exist based on antibodies that can multiplex the detection of, say, 5,000 proteins. It's very costly. It's not high throughput. But there's so much more that we can learn from proteomics than we do today, and especially large-scale proteomics. And that goes beyond cancer. So with cancer, most of the aberrations are based in DNA. Cancer is a disease of the DNA. So we can detect mutations and other kinds of events that occur in DNA. Mutations, epigenetic modifications, etc. All of that occurs in DNA. We can see that with sequencing, both DNA sequencing and RNA sequencing, but we don't have a ton of information on proteins. And in cancer, this is going to help, but it's also going to help in other diseases.
Great. Please go ahead.
I really liked your question, actually. But my question was, in the cases where the GPS AI model is not correct, what are the things that are driving the misclassifications?
Oh, that's hard. What's driving the misclassifications is, so sometimes the quality of the data. So when we're dealing with extremely small tumors, there's so little material that it's hard to generate sufficient sequencing data to generate the proper calls. So some of those misclassifications we actually know from the beginning that sadly there's not enough. There's also some of the misclassifications in the cases where we have mixtures of multiple cell types due to the location of the tumor. So I showed some of the confusion that could exist for primary samples that have metastasized to another organ. That can generate some confusion. So that could be some of the misclassification.
Thank you.
All right. So we'll take a short break now, but do come back at 11 for our next talk.